bioethics,
law, euthanasia,
consumerism,
politics, terrorism,
globalization,
religion,
migrant population,
interfaith, drug addiction.
In a youth town hall meeting in 2010, US President Barack Obama was asked if he thought gay or transgendered people have a choice or are born that way. According to a CNN report, the president replied, "I don't think it's a choice. I think people are born with a certain makeup." Most Americans seem to agree with their president. According to the results of a new Gallup poll published on the 20th of May 2015, the majority of Americans believe that gay and lesbian people are born, not made. Over 51% of those who participated in the survey maintain that same-sex orientation is genetic, a matter of birth, and not the result of upbringing or the environment. We should not think that this is just the opinion of lay people. A number of prominent scientists and medical professionals also believe that homosexuality is biological or innate. And they include the neurologist Simon LeVay, the director of the Virginia Institute of Psychiatric and Behavioral Studies, Kenneth S. Kendler, and the former head of the Department of Psychology at Queen's University, Vernon L. Quinsey. In this talk, I will examine some of the most important studies that have been conducted over the past 20 years to see if they show conclusively that homosexual orientation is determined solely by biological factors like the genes or the brain. I will evaluate the Kinsey proposals on sexuality in general and homosexuality in particular. I will also discuss the circumstances that led the American Psychiatric Association to change their diagnosis and status of homosexuality and examine if this change is warranted by conclusive scientific research or propelled by political pressure from gay activists. And finally, I will also discuss the various reparative therapies and evaluate their effectiveness in helping people with same-sex attraction. So that's the outline, that's the agenda for this talk. We turn our attention firstly to the twin studies conducted by John Michael Bailey and Richard Pillard in the late 1980s. Bailey was an American psychologist at Northwestern University, and Pillard was a professor of psychiatry at Boston University. The purpose of the twin study by Bailey and Pillard is to ascertain whether in a pair of twins, if one twin is homosexual or has homosexual tendencies, the other twin would also have similar tendencies. Bailey and Pillard studied 54 sets of monozygotic twins and 44 sets of dizygotic twins. In a paper published in 1991 entitled A Genetic Study of Male Sexual Orientation, Bailey and Pillard reported that they have discovered a striking concordance rate of 52% among identical twins and 22% among non-identical twins. This means that among the 54 sets of identical twins, half of the pairs were homosexual, while for the other half, one twin is homosexual and the other is heterosexual. And since identical twins have the same genetic makeup, Bailey and Pillard concluded that the high concordance rate simply shows that homosexual orientation must have a genetic basis. Now, it is important to note that the study by Bailey and Pillard has to do with statistics. They have not discovered, nor have they claimed to have discovered the gay gene. But because their study took place in a context where there is so much hype about genetics, both the public and the media accepted their conclusions uncritically. A number of issues can be raised concerning the assumptions and methods in the twin study, especially the way in which the subjects for that study were recruited. Some critics have noted that the study rests on the assumption that the social experiences of both the homosexual twins were similar. However, if this is not in fact the case, then environmental contributions to the homosexual proclivities of the subjects of this study must be taken into serious consideration. As I mentioned earlier, the way in which the participants of the research were recruited has also been heavily criticized. Bailey and Pillard recruited their research subjects by placing an advertisement in a gay magazine. As Jeffrey Santinova has rightly pointed out, a common problem in these kinds of studies is that concordant twins tend in general to respond to research advertisements more frequently than twins where one is homosexual and the other heterosexual. Readers of homosexual magazines, he adds, are in no way representative of homosexuals. In 1992, Michael King and Elizabeth MacDonald conducted a similar study in Britain. In their paper entitled Homosexuals Who Are Twins, a study of 46 probands, King and MacDonald reported a much lower concordance rate for homosexuality compared to the study conducted by Bailey and Pillard. There is a concordance rate of only 25% among identical twins compared to the 52% in the Bailey and Pillard study. And among non-identical twins, King and MacDonald reported a 12% concordance compared to 22% in the Bailey and Pillard study. Now, these findings have led King and MacDonald to arrive at a very different conclusion to Bailey and Pillard. And so they state in their report, this concordance for sexual orientation in monozygotic pairs confirmed that genetic factors are insufficient explanation for the development of sexual orientation. Now, even if we accept the concordance rate reported by Bailey and Pillard, we must still challenge their conclusion. As we have seen, based on their findings, Bailey and Pillard concluded that there must be a genetic basis for homosexual orientation. This conclusion, however, is somewhat puzzling because the results seem to indicate that the environment played a significant role in homosexuality. If homosexual orientation is in fact determined by genetic factors, that is the result of having a particular genetic code, then why is it that not all the identical twins in the study homosexual? Since identical twins have the same genetic makeup, why is there only a 50% concordance and not 100%? As Jeffrey Santinova has rightly pointed out, contrary to what Bailey and Pillard had suggested, the findings should lead to the conclusion that homosexuality is influenced by non-genetic factors. This finding alone argues for the enormous importance of non-genetic factors influencing homosexuality, writes Santinova. Because in order for something to be genetically determined as opposed to merely influenced, the genetic heritability would need to approach 100%. Many critics of the twin study by Bailey and Pillard concur with Satinova's assessment. So for example, William Bine, the director of the Laboratory of Neuroanatomy and Morphometrics at Mount Sinai School of Medicine, argues that the study clearly challenges the simple genetic hypothesis and strongly suggests that environment contributes significantly to sexual orientation. In their article in Technology Review entitled Born Gay with a question mark, Paul Billings and Jonathan Beckwith note, while the authors, that is Bailey and Pillard, interpreted their findings as evidence for a scientific basis for homosexuality, we think that the data in fact provides strong evidence for the influence of the environment. So far from demonstrating convincingly that homosexual orientation can be attributed to the genes, the study conducted by Bailey and Pillard in fact points to a more complex picture, one in which the environment and the experiences of individual subjects play an important, if not critical, role. Bailey himself realized that his original study was not representative, and so he conducted a follow-up study with MP Dune and Angie Martin in the year 2000, this time taking the sample from the Australian Twin Registry. In this second study, Bailey reported a much lower concordance rate, 20% for identical twins, compared to 52% in his earlier studies. In other words, the result of Bailey's second study correspond more closely to the findings reported by King and MacDonald. In 2010, an impressive and large-scale study was conducted using the Swedish Twin Registry. The concordance rate among identical twins reported in their study is stunningly low, at only 9.8%. So clearly, the difference concordance rates points to the important role that non-genetic factors like the environment, life experiences, and relationships play in homosexual orientation. We turn next to a study conducted by Dean H. Hammer and his team of researchers at the National Cancer Institute in the United States in 1993. Unlike the study conducted by Bailey and Pillard, which has to do with statistics, the Hammer study sought to identify the biological basis of homosexuality by analyzing the chromosome of homosexual men. Working with 40 pairs of homosexual brothers, Hammer and his team discovered that 33 out of the 40 pairs, or 83%, have the same sequence of markers in the X chromosome region known as XQ28. In a paper published in 1993 entitled "A Linkage Between DNA Markers on the X Chromosome and Male Sexual Orientation," Hammer states that one form of male homosexuality is preferentially transmitted through the maternal side and is generally linked to chromosomal region XQ28. Now, the press immediately publicized this discovery by churning out articles with seriously misleading titles. So, for example, the New York Times has a front-page article entitled "Report Suggests Homosexuality is Linked to Genes." The Wall Street Journal headlined their report with a title, "Research Points Toward a Gay Gene." Although the content of the article was more nuanced, their sensational titles captured the imagination of the public, leading many to think that the genetic basis for homosexual orientation has at last been found. Hammer's study was fraught with serious problems associated with methodology and problems associated with sample size. One critical problem with the study is that there is no control group from the general population. So, for example, Hammer did not test the results against a heterosexual control group. If the same sequence that appears in the chromosomal region XQ28 of the homosexual men is also found in the heterosexual population, then the presence of that sequence will be inconsequential. Furthermore, Hammer did not test the heterosexual brothers of the homosexual men to see if they also have the same gene. 4 months after Hammer published his paper in the magazine called Science, critical commentaries began to appear in that same publication. So, for example, a paper entitled Male Sexual Orientation and Scientific Evidence, published in the December 1993 issue of Science, Neil Risch and his co-authors state that Hammer's study is not consistent with any genetic model. Neither of these differences between homosexuality in maternal versus paternal uncles or cousins is statistically significant. On June the 25th, 1995, reports surfaced that Hammer had come under investigation conducted by the National Institute of Health's Office of Research Integrity due to allegations that he ignored or suppressed evidence that contradicted his hypothesis. Although NIH did not in the end publish the results of the inquiry, Hammer was transferred to another department. In April 1999, George Rice and his associates tried to replicate Hammer's study by examining the same marker in 52 gay sibling pairs. They concluded, in agreement with Neil Risch, that the marker in question is statistically insignificant. In a report published in Science in 1999, they wrote, "Our data does not support the presence of a gene of large effect influencing sexual orientation at position XQ28. In response to these criticisms, Hammer, who is an openly gay man who got married to his partner, longtime partner, in 2008, made the following qualifications about his conclusions. He wrote, "We did not say that XQ28 underlies sexuality, only that it contributed to it in some families. Nor have we said that XQ28 represents a major gene, only that its influence is statistically detectable in the population that we studied." In the same article, Hammer admits that it is problematic to ascertain the significance of the statistics he had obtained from his study in relation to a trait as complex as sexual orientation. And so he writes quite honestly that the question of the appropriate significance level to apply to a non-Mendelian trait such as sexual orientation is problematic. Hammer was therefore never as sanguine about his findings as the media and the gay activists had exaggerated them. In a book co-authored with Peter Copeland entitled The Science of Desire, published in 1994, Hammer states, again quite honestly, that the pedigree study failed to produce what we originally hoped to find: simple Mendelian inheritance. In fact, we never found a single family in which homosexuality was distributed in the obvious sort of patterns Mendel observed in his pea plants. In the same book, Hammer admits that the role played by the environment in human sexual orientation simply cannot be ignored. He writes, We knew also that genes were only part of the answer. We assumed the environment also played a role in sexual orientation, as it does in most, if not all, behaviors. To most people, the environment means non-biological factors such as family upbringing, life experiences, and religion. In 2014, Michael Bailey, the same Michael Bailey of the twin study fame, and Alan Sanders of North Shore University Health System conducted a study along similar lines taken by Dean Hammer 20 years ago. And he conducted this study on 409 pairs of brothers, making it the largest study to date. They were surprised to find the same linkages between homosexuality and the chromosomal region XQ28 that was suggested earlier by Hammer's study. Hammer was naturally delighted with the result. 20 years is a long time to wait for validation, he reported to have said. But now it's clear the original results were right. It's very nice to see it confirmed. Equally pleased is the neuroscientist Simon LeVay, whose 1991 study we shall be turning to very shortly. LeVay reported that this study knocks another nail into the coffin of the chosen lifestyle theory of homosexuality. What are we to make of this? Does this mean that scientists have finally found the genetic determinant for homosexual orientation? Well, the answer is no. Although the Sanders study is significantly larger than the one conducted by Hammer, the findings fail to conclusively identify a gene or a cluster of genes responsible for determining sexual or homosexual orientation. Behavioral geneticists have found the results of the Sanders study unconvincing. The method that Sanders and his associates used to establish genetic linkage have been superseded by other techniques, for example, by the Genome-Wide Association, or GWA. Sanders himself acknowledged that GWA is superior to linkage studies, but he felt that he had to use linkage studies in order to replicate Hammer's original study. Neil Risch, who criticized the Hammer study, also found the findings of the Sanders study to be statistically insignificant. In fact, even Sanders himself admitted that the linkage to XQ28 in his study has failed to cross the threshold of significance as delineated by standard scientific practice. And although he believes that the chromosomal region XQ28 may have something to do with homosexual orientation, Sanders insisted that complex traits like sexual orientation always depends on many factors, both genetic as well as environmental. In many ways, these attempts to discover the genetic basis for homosexual orientation have repeatedly confirmed what many scientists, philosophers, and theologians have known for a long time. The distinction must be made between the genotype, which is the set of genes in our DNA that can be associated with a particular trait, and the phenotype, which has to do with the expression of that trait in that organism or in that subject. The relationship between the genotype and the phenotype is not always clear and mostly never straightforward. And that is why scientists have always cautioned against a form of genetic determinism, which in fact collapses the phenotype into the genotype, ignoring non-genetic factors. In fact, the general consensus is that the genotype always underdetermines the phenotype. John Maddox expresses this very well when he writes, "The link between genotype and phenotype is not always unambiguous. A genotype may be a necessary, but not a sufficient condition for the phenotype." The relationship between gene expression, the environment, and behavior is so complex that a new field has emerged called epigenetics, which focuses its study on this relationship. Epigenetics looks into the way in which genes react to their own immediate physical or cellular environment, as well as the way in which they respond to external environment and the life experiences of the organism or the subject. Genes have the ability to switch on and off as circumstances dictate. And many have found that gene expression and behavior are especially influenced by early social experiences. Social experiences throughout life influence gene expression and behavior. However, early in development, these influences have a profound effect, writes Francis Champagne of Columbia University. This means that many important questions are still left unanswered by the study conducted by Sanders. If there is in fact a link between XQ28 and sexual orientation, and I must emphasize that this is a very big if, to what extent does it influence or determine sexual behavior? How do the other genes in the immediate environment interact with this gene in question? And what about the external environment? How might this have contributed to sexual preferences and behavior? Sanders himself acknowledged the gravity of these questions. So in an interview conducted by LGBT Science, an organization dedicated to exploring the origins of sexual orientation, Sanders quite honestly said, "When people say there is the gay gene, it's an oversimplification. We don't think there is just one gene involved. There are a number of genes. We also don't think genetics is the whole story. It is not." Now, while Hammer, Sanders, and their associates focused on the genetic determination of homosexual orientation, others have turned to neuroscience to see if sexual orientation is somehow hardwired in the human brain. Simon LeVay of the Salk Institute for Biological Studies in San Diego conducted an important study that aims to discover if there is a neurological basis for homosexual orientation. LeVay, who is an openly gay man, studied the brains of two groups of men. The first group, homosexual men. The second group, men he presumed to be heterosexual. He claimed to have discovered a subtle but significant difference in the brains of homosexual and heterosexual men that may be responsible for homosexual orientation. LeVay focused his study on the area of the brain called the hypothalamus. This is the section of the brain that is responsible for the production of the body's essential hormones that control the different cells and different organs. The hypothalamus also governs important psychological functions such as thirst, hunger, sleep, sex drive. LeVay then narrowed his focus on a cluster of cells in the hypothalamus called the third interstitial nucleus of the anterior thalamus, or INAH3. He found that this cluster of cells is smaller in homosexual men than in heterosexual men. And he concluded that this has significant bearing on male homosexual orientation. And as to be expected, the media hastily announced that homosexuality is hardwired in the brains of some men. There were, however, serious problems with LaVey's study. In the first place, he worked with a very small sample, only 19 homosexual men and 16 allegedly heterosexual men. The claim that the second group comprises solely heterosexual men has been called to question. And LaVey, under pressure, admitted that he was not entirely sure if all the men in the second group were heterosexual because he did not investigate their sexual histories. He could not investigate their sexual histories because he was examining cadavers. In addition, 6 of the heterosexual men died of AIDS. 6 of the heterosexual men died of AIDS. And given the statistical fact that very few heterosexual men in San Francisco contracted AIDS in the early 1990s, it is not improbable that some of the men in LaVey's second group were either homosexuals or bisexuals. The brains of these men, as I mentioned earlier, were examined post-mortem, which explains why it is impossible to obtain their sexual histories.